Good morning, everyone. Welcome to the short oral presentation, section 1. Okay, my name is Suyanee Thongchot from Department of Immunology and as I call Cate, Chinsuwan Hospital, Nithidol University. And I have Dr.
I'm Thanyavi Chinsuwan from Cell-derived Immunotherapy Research Unit, Chulalongkorn University.
Okay, so we will start with our first presenter. Dr. Kajornkiat Maneechai. He will give a talk in the title of the generation of autologous patient hematopoietic stem cell derived the T-cell T-lymphocyte for adoptive T-cell therapy. So everybody, please help me to welcome.
Thank you very much for kind introduction. Actually, I was the master degree candidate. So a very good morning to all of you again. So first, I would like to give an appreciation to TTCI and everyone here for the warm welcome again. I'm Kajornkiat from Songkhla University, and here's the research for today. So first of all, I would like to give the introduction about this study. As you see in this figure, this is the magnification inside the bone marrow where the hematopoietic stem cell is located, which is the cell that enables to differentiate into the archive of the mature blood cell over here. According to this property, it's leading to stem cell transplantation process that consists of the cell collection, chemotherapeutic exposure, and the infusing back of the stem cell for the blood cell regeneration. In addition to stem cell transplantation, another treatment of choice is adoptive cell therapy, which is the way to teach our immune cell to fight against the cancer. And now we are focusing on the chimeric antigen receptor that anomaly talked on the period yesterday and today. And as I mentioned earlier about the chemotherapy, this is the table that demonstrates the repetitive chemo exposure on both CD8 and CD4 T cell that cause the impairment of T cell function and lower of T cell population. That's the limitation of the ACT, that we cannot collect the mature T cell from the patients. To solve this problem, that's why we need to find the other sort. The one is from the allogenic healthy donor hematopoietic stem cell that can use as the starting material to generate a mature T cell in vitro. So this leads into the previous study that many researchers here try to generate the mature T cell from the umbilical cord blood hematopoietic stem cell with the various steps and many of the cytokine supplementation. However, the outcome of this study, they got just only the CD8 single positive T cell, which is not suitable for the setting of adoptive cell therapy. So in this study, we aim to generate the mature T cell from the patient peripheral blood with the optimized the generation protocol. So this is the generation procedure that divides into 3 phases. The first one is progenitor T cell differentiation from day 0 to day 14. The second one is a double positive T cell differentiation from day 14 to day 42. And the last one will be the single positive T cell maturation further for 10 days. And the figure below here demonstrates the expected the biological process chain at each time point of the differentiated cell. For the first result, this is the cell isolation on day 0. In this figure demonstrate the representative flow plot. On the x-axis is show the CD3 and the y-axis shows CD34. And we could isolate it around 90% of both CD3 and CD34 over here. And for summary, in 6 patients, it show around 91% and 91% and 86% respectively. For the next result, this is the representative flow plot again that shows the various marker molecule on the x and y axis. And we can see that the dynamic of the cell proportion at each time point. For example, in this quadrant over here, this is show the population of the CD5, CD7 double positive cell, which can identify as the progenitor T lymphocyte. So this show around 40% on day 14, but and it's increased on time to around 80% in the 52, as well as the expression of the CD3. And here's the summary for 5 patient donors that include the lymphoma and multiple myeloma, which they got the difference of the chemotherapeutic exposure. And you can see that some donor cell could not differentiate into the mature T cell. And this is a summary bar chart, and each color here demonstrates each time point. And you can see that the CD45 keep rising on day 28 and maintained to day 52, as well as the expression of the CD5, CD7. And importantly, we observe around 90% of the CD3 at the day 52, with the most of the cell with 2 fractions of the CD8 and 1 fraction of the CD4 here. And this is the full expansion of the differentiated cell from day 0 to day 52, and we achieve around 3,735 full expansion. For the next one, this is the T cell receptor, which is the maturity receptor of the T cell. So we assess the expression of this kind of receptor. So we stay in this cell and perform the flow cytometry, and it shows around 90% expression of the alpha beta T cell receptor of the conventional T cell. In contrast, the HSCT or the derived cell shows just around 50%. So this result could be explained by the lack of the thymic cortical epithelial cell, which normally are located in the thymus gland. For the T cell immunophenotype, in terms of the stemness, the phenotype of the derived cell show the naive as the majority. In contrast of the conventional T cell, they show the distribute into 4 subpopulation over here. For the T cell exhaustion, we observe the no significant difference of the exhaustion marker. However, the TIM3 molecule seems to be higher in the HSCT. And for the assessment of the cytokine secretion, we observe that the HSCT show the greater trend of the cytokine secretion, including interferon gamma, interleukin 2, granzyme beta, and perforin. And this is the application in this study by generating CD19 CAR T cell by using the in-house structure from our lab. And the protocol was reviewed in the figure B here. For briefly, it was the retroviral transduction. And the results show around 50% of the transduction efficacy and more than 90% of the purification. And furthermore, both of the CD19 CAR T cells show the similar fold expansion in the figure B here. Figure D, sorry. And for the next, we assess the proliferative capacity of the CD19 CAR T cell, and we found that both of the HSCT and conventional CAR T cell show the similar fold expansion with in the condition with or without interleukin 2 supplementation. And for the next result, this is a specific cytolysis against the primary mantle cell lymphoma on the left panel here. And it showed that the HSCT CAR T cell showed the superior cytotoxicity against the, compared to the conventional CAR T cell. For the NAM6 cell line, it showed the similar cytotoxicity. And lastly, we performed 3 consecutive weekly stimulation to assess the fold expansion and phenotype. Both of the CAR T cells show no difference of the fold expansion. And in addition, both of the CD18 CAR T cell immunophenotype and exhaustion markers show the incomparable result. So the discussion this study, we successfully generated T lymphocyte from patient autologous hematopoietic stem cell with preserved cytotoxic T cell function compared to the conventional T cell. The second one is there were the variation of the T cell function among the donor because of the nature of the underlying disease and also the difference of the chemotherapeutic regimen. For the third point, the alpha beta T cell receptor demonstrate only 50% of the HSCT population. So this result might be the consequence of the lack of the thymic cortical epithelial cell, as I mentioned earlier. And for the limitation in the study that small sample size and only multiple myeloma and lymphoma patients were included, so we need to do further enrollment for the large scale for the experiment. This is my team and acknowledgement. Thank you very much for your attention.
Thank you very much for your nice presentation. I really like your idea because we are frequently facing the difficulty to collecting lymphocytes upon manufacturing CAR T cells, both Kymriah and also our CAR T products. And my question is, have you ever tried to generate CAR T cells from your T cells, HCT derived T cells? Have you ever tried?
Yes, is the work. The work of the study is to.
Sorry, I missed it. Okay. So sorry. Okay. So sorry. Yeah. I'm okay. Thank you.
Okay. Any more questions from the floor? All right, Professor Hashio.
Thank you very much for your talk. So you showed the TIM3 high expression on the HSCT, right? TIM3. So I thought that was interesting because Shoji and Shigeki previously showed their piggyback CAR. It has quite a lot of naive memory stem cell population, and your HSCT also had that population. And their CAR also had high TIM3. I didn't know why, and do you have any idea why your HSCT had high TIM3 expression?
Okay, thank you very much for the question. There's interesting question here. So at first, in my opinion, I think this result is the consequences of the protocol that we generate the HSCT, because on the day 52 we got some kind of the lymphocyte. We can verify that is the lymphocyte. Visually, it is lymphocyte. So we activated that cell with the CD3, CD28, the microbead. So this experiment may lead to the stimulation of the cells. So the TIM3 will be like the TIM3 marker could be like expressed more with this experiment after actuate with the bead.
Okay, we have to move to the next speaker. So thank you.
Next speaker, we have Kristine Cate from our CIT research unit. So take it away, Kate.
So before I start, I would like to say, that I'm very grateful. This is too tall for me. I'm very grateful that I get to present in front of all these many experts in our field. But I'm also truly terrified that I get to present in front of all these many experts in our field. Okay, John, let's start. So good morning again. My name is Kate. I'm here to present my work on assessing the influence of non-signaling domains on TIM3 CAR T cell development for the treatment of AML. Okay. So we all know the design of CAR T cells include ligand binding domain, spacer domain, transmembrane domain, and the cytoplasmic domain. And researchers have been investigating several potential strategies to improve CAR function and its persistence. And they have tried to modify CAR. Several efforts have focused on the antigen recognition domain and the cytoplasmic signaling domain. So a domain in the CAR construct that is usually overlooked in evaluating CAR functionality is the non-signaling spacer and transmembrane domain. So the choice of spacer and transmembrane domain in CAR T cell can significantly influence CAR function. A spacer domain aids in the accessibility of the CAR to recognize target antigen, where the long spacers provide better access to membrane proximal epitopes, while the short spacers are highly effective to membrane distal epitope. The transmembrane domain regulates the amount of CAR signaling via the control of CAR expression level, and this domain has been known to influence cytokine release, which, if excessive, may lead to high unspecific toxicity. The transmembrane domain, along with the spacer, can affect the CAR signaling intensity into T cells. So here, FDA-approved anti-CD19 CAR T cells, namely the axi-cell, T-cell, and the lyso-cell. The success of this CAR T can be attributed to their antigen specificity, all conferred by the scFv of the anti-CD19, and their different intracellular signaling domain. It is also worth noting that these products differ in their spacer domain and spacer domain or the hinge domain and their transmembrane domain. That is CD28 hinge domain and transmembrane domain for axi-cell, CD8 hinge domain and transmembrane domain for T-cell, and IgG4 derived hinge domain and CD28 transmembrane domain for lyso-cell. So different combination of non-signaling domain have their own pros and cons. CD28 have resulted in heterodimers in combination with either a CD8 or CD28 spacer in CAR T cells. This resulted in reduced expression of the surface CD28 and may result to impaired CAR T cell, as it increases susceptibility to T cell exhaustion and differentiation to an effector memory phenotype. However, when CD28 transmembrane was paired with an IgG-derived spacer, it resulted to an inefficient heterodimerization. In another study, the combination of the CD8 and the CD28 resulted to CAR T cell tonic signaling due to increased expression of the CD25 and the PD-1. This tonic signaling was diminished by replacing CD8 with an IgG-derived spacer. And the combination of the CD8 spacer and transmembrane is successfully used in BCMA-directed CAR T cell for patients with multiple myeloma. So recently, TIM3 was identified as a surface molecule expressed in leukemic stem cells of AML. The group of Kikoshige reported that there's a very low expression of TIM3 on hematopoietic stem cells on this figure. And a very high expression of on AML leukemic stem cells and bulk AML. Another group reported the same result and found that TIM3 is positively expressed on leukemic stem cells where normal hematopoietic stem cells completely lack TIM3 expression. Therefore, TIM3 is a promising target in AML as it can be used to separate normal hematopoietic stem cells from AML leukemic stem cells and also found on AML bulk. So for this project, we want to generate two CAR T cells that both targets the TIM3, with one CAR consisting of the CD8 spacer and transmembrane domain, while the other CAR consists of the IgG-derived CH3 spacer and CD28 transmembrane domain, and the intracellular signaling for both CAR will consist of the 4-1BB and CD3 zeta. So we first screened for the expression of TIM3 on normal hematopoietic cells, wherein we found that TIM3 is expressed highly on activated T cells and very low expression on NK cells and monocyte, and also absent on B cell and resting T cells. We screened for 37 AML patients and found that TIM3 is expressed with an upper limit of 83.5% and lowest expression with 0%. TIM3 was also expressed in THP1, Kisuми1, and SKM1 AML cell lines. So we then generated our anti-TIM3 CAR T cells, one with the CD8 non-signaling domain and the other one with the CH3 CD28 non-signaling domain. So upon analyzing the percent CAR expression, CH3 CD28 resulted to a very low expression in T cell surface compared with the CD8 on day 4 and day 11. When we checked the immunophenotype of our CARs, both non-signaling domain resulted with a 1-to-1 CD4-CD8 T cell subset, and both were predominantly consist of the naive T cells. We also assessed for T cell proliferation from day 4, 7 to 11 and found that both CAR T cells were proliferating exponentially, where the CD8 non-signaling domain proliferated more than the CH3 CD28. And because activated T cells also express TIM3, we decided to determine the extent of fratricide by screening first for the expression of TIM3 on our CAR, and for the apoptotic cells on day 4, 7, and 11. We found that there was lower expression of TIM3 on CD8 and almost none on CH3 CD28 CAR. CD8 non-signaling also resulted to enhanced fratricide, while low level of fratricide was detected with CH3 CD28. The fratricide activity on both CAR decreased eventually till day 11 and did not interfere with CAR expression, and the cell viability remains to be more than 80%. So lastly, we perform comparison of the anti-tumor activity between the non-signaling domain. We co-culture our CAR T cells with 3 AML cell lines, SKM1, Kisumi1, and THP1, and use K562 as the negative target. Both non-signaling domain exhibited dose-dependent tumor killing while sparing the negative target. The anti-TIM3 CAR with the CH3 CD28 non-signaling domain can kill better compared with the CD8 CAR. However, cytokine secretion were on the same level for both CAR with an elevated interferon gamma release. So to conclude everything, in comparing the two non-signaling domains, CD8 resulted to a higher CAR expression and improved cell proliferation, but both non-signaling domain resulted to fratricide during in vitro expansion. Nonetheless, the absolute T cells continually increased with both of the non-signaling domain. CH3 CD28 resulted to a higher tumor killing compared with the CD8 despite the CAR expression differences. And our results demonstrate that Non-signaling domain contributes to anti-TIM3 CAR functionality, and anti-TIM3 CAR with CD8 alpha non-signaling domain have the potential clinical advantages due to its CAR expression and expansion. That will be the end of my talk. I would like to express my deepest appreciation to my advisors, Kajornkiat and Kajornkiat. And I'm also grateful to Ali and PUE. Lastly, thank you to Chulalongkorn University ASEAN Scholarship. Thank you.
So, okay.
Hi, Kate. Thanks.
Hi, Ali.
Lovely talk. I'm just wondering, with you and some of the other groups using CH2, CH3 domains, did you mutate the residues that impart FC binding?
Actually, for this work, we just used the CH3. We didn't use the CH2, CH3 sequence. So for this one, we use the IgG with only consisting of the CH3. So we did not observe any toxicity with any. So we expect to not actually observe any toxicity with normal cells.
Yeah, it's just this negative impact of the CH2, CH3 binding to FC receptor domains on adjacent cells, monocytes and NKs and so forth.
So from the paper of Watanabe, they tried to compare different IgG and also try to modify the CH2, CH3. CH2, CH3 would definitely bind to monocyte and other immune cells. But when they tried to remove the CH2, only having the CH3, they didn't see any cytotoxic against immune cells. And if you use the IgG2, which we actually use in this one, these have the lowest binding affinity compared to other IgGs.
Good answer, Kate. I give you 10 points.
Thank you.
Hi, thank you very much for your beautiful talk. And you clearly mentioned that the acceptable fratricide during the manufacturing step, but TIM3 can be induced after the antigen expression because TIM3 is the immunocheckpoint protein. So did you check the proliferation or fratricide after the antigen simulation?
Oh, that part, actually, no, we didn't get to check if the cytotoxic is on the fratricide or the killing of CAR to the TIM3. So this one is we just checked the short-term co-culture of our CAR against target antigens in AML.
Thananyai. She will present the work in the title, Development of a novel anti-B7H3 antibody for designing CAR T cell against solid tumor. Everybody, please.
Okay, thank you for your welcome. My name is Thananya Inthanachai from Kajornkiat University. I'm working on the development of a novel anti-human B7 homolog T antibody for designing CAR T cell against solid cancer. So B7H3 is an adaptive antigen. This molecule plays an important role in down-modulating lymphocyte activation and inducing cancer glycolytic metabolism. The expression of B7H3 was found to upregulate exclusively on the cancer cell and show a positive correlation with poor clinical outcome. Recently, it has become a useful marker for detecting tumor and metastasis in many cancer types. So, to highlight that B7H3 is a common antigen overexpressing across various types of cancer, we analyzed an RNA transcription level from TCGA and GTEC database. Our finding revealed that among 29 solid cancer types, the CD276 gene exhibits a significant upregulation across 16 types of cancers. We also confirm an expression level on cancer cell surface using the flow cytometric staining. We found B7H3 highly expressed on many different solid cancer cell lines, including ovarian, breast, bile duct, and glioblastoma, with a very mean fluorescent intensity to isotype, as shown in the number here. These results support a potential use of B7H3 as a target surface antigen for solid cancer treatment. So, to develop B7H3 CAR T cell from our new antibody for treatment solid cancer, we divide our work into three main parts. Firstly, after immunization mouse with the human B7H3 protein, we obtain 4 hybridoma clone producing monoclonal antibody that selectively bind to human B7H3. Their affinity were measured by surface plasmon resonance using the Beer-Core T200 and show as equilibrium dissociation constant or KD value in this table, range from -8 to 10 molar. To test their binding on tumor surface antigen, we stain B7H3 positive cancer cell with this novel monoclonal antibody and analyze through flow cytometry. In this figure, the novel antibody clone 7A5 show a non-specific binding with NAM6, which is a B7H3 negative cell, but the clone 12G5 and especially high affinity clone 11G5 and 2H4 can show specific binding only to B7H3 positive cell line. Therefore, we select this T monoclonal antibody to develop into the scFv format in CAR construct and together compare with the published clone T7696 for a positive control as shown in this schema here. 4 days after retrovirus transduction, we found that the truncated CD19 as a surrogate marker to check transduction efficiency can be similarly detected on T cell across all constructs. Interestingly, when we use B7H3 protein to bind directly with the scFv in the targeting domain, we can detect different expression between each construct. This variation was related with the affinity of monoclonal antibody that they derive from. As you can see, 11G5 and 2H4 CAR higher expressed than 2G5, which is the lowest affinity in this development. This indicating that new B7H3 specific monoclonal antibody can develop into CAR T cell, but intrinsic affinity of monoclonal antibody can individually affect on capacity of antigen interaction. With this instead, we evaluate more detail in their characteristic. We found a higher ratio of effector memory and central memory T cell in the 12G5 and 2H4 CARs that could benefit them in the long-term persistence than others. However, the difference in the 4 expansion, T cell subset, and exhaustion will not occur. Next, we test anti-tumor activity of each CARs with exogenous real-time analysis. The results show that none of B7H3 CAR T cells exhibit anti-tumor activity against NAM6, which is a B7H3 negative cell lines, but all efficiently against LN229, which is a B7H3 positive glioblastoma cell. With this activity was observed in a dose-dependent manner, and there were no significant difference between the anti-tumor effect of each CAR construct. Along with the cytokine result, IL-2 interferon gamma and TNF-alpha highly secrete only upon antigen stimulation, especially by 12G5 and 2H4 CARs. Besides, to mimic tumor microenvironment, we set our LN229 in the 3D and co-culture with effector cells. Using the IncuSite, we continually observe the intensity of green object from GFP tag target cells and red from effector cell. Surprisingly, even 12G5 CARs having the lowest affinity binding to the antigen, we notice more rapid infiltration into spheroid of this CAR. Moreover, it demonstrates a significantly better functionality against glioblastoma spheroid than the other construct. This result leads to the conclusion that we succeeded in generating new monoclonal antibody that's specific to human B7H3 and were able to bind to many different solid cancer cell lines. Moreover, CAR T cell development from this specific antibody can provide a specific function against solid cancer expressing B7H3 and trained to perform with the different potency. Therefore, to pick up the best candidate, B7-H3 CAR T-cell function will evaluate more in vivo for application in clinical translation and future development. Finally, I want to give my sincere acknowledgement to my PI, all CUAC, CUIT, and CUSB lab members for helpful advice and support. Also, thank you all of you for attention.
Thank you, Thananya. Any questions from the floor?
Really nice talk. Congratulations on making so many antibodies. You're using an FC domain to immunize the mice? I missed that. You used an FC fusion protein to generate those. Did you get a chance to compare them to some existing B7H3 CAR?
Yeah.
No, no. Okay, we could probably.
Oh yeah, the public sequence one.
Where's the existing one? Oh.
Yes, public sequence CAR.
Which one's
The T7696. This is a positive control. Yes.
Thank you for your excellent talk. I'm curious, like, can you go back to the previous slide that showed the KD?
The KD.
Right. So you mentioned in the almost last slide that the antibody clone that show are not good, like low affinity with this KD, but show very good toxicity, going to kill the cells more than the high affinity. What is the mechanism of that effect?
Yeah, it's for this reason. I think it might be because the kinetic of this antibody, because the kinetic, the graph, the former graph of this monoclonal antibody, it seems to that this monoclonal antibody can bind with the target quickly, and then they leave the target quicker than other construct. So it might be effect on the in the spheroid that when they quickly attack to the target, they kill, and then they just quickly to leave the target and kill to another cell. So that might be effect to the better function to another construct, in my opinion. Yeah.
Hi, my question is simple. Are those antibody recognize different sites on the target molecule? Do you know where the antibody recognize on the B7H3?
Yeah, for the site of the binding, we didn't investigate it yet, but it's so interesting to perform, but it will take a lot of money. Yeah.
Because that may be affecting the result of the preparation.
Thank you.
All right, I guess that's all we have time for. Okay, seeing one more question.
Thank you for an impressive talk. I have also the simple question about the antibody. So as I understand correctly, so this SCFV is from mice.
Yes.
So they mostly recognize mice. Is it affect the binding to the human target?
Yeah, for the effect to human B7H3, it might be effect, but when we measure the affinity, we use the human B7H3 to measure that. So the result that we show is the real value that affect to the human B7, yes, not mice.
And one more question, when you transfer from antibody to CAR T cells, do you need to humanize this SCFV to use it in humans or not? Because for antibody, you should humanize it.
Oh yeah, but for this, for our experiment, we didn't perform the humanize to do the CAR construct. We just use the sequence that we found and then developed into the CAR construct.
Okay, okay, thank you.
Thank you.
All right. Thank you, Thananya, for your excellent answers. For the final presenter, we are going to have Nithidol Sakunrangsit presenting for us. So he is going to talk about development of a chimeric antigen receptor targeting folate receptor alpha from human variable domain of heavy chain antibody fragment. So please give a round of applause for Dr. Nithidol.
Good morning, everyone. I'm very happy to be here to share my research in this topic. As you know, CAR T-cell therapy has emerged as a promising treatment option for cancers. Nowadays, many clinical trials are ongoing for both hematologic malignancy and solid tumors. In general, single-chain variable fragment or scFv derived from monoclonal antibody are common use for ligand binding mediated tumor cell recognition in CAR T-cell therapy. scFv is derived from the variable domain of heavy and light chains of antibody connecting the VH and VL with linker. The size of scFv is usually ranging from 800 to 900 base pair, whereas VH antibody is smaller size that is derived from variable domains of heavy chains only fragments. This line shows the example of VH-based CAR T cells. VH antibody have many benefit properties, include their small size that allow them easier access to tissues, high stability, strong affinity to their cognate antigens, and low immunogenicity, all of which make them very good candidates for development into next generations CAR T-cell products. VH antibody are useful alternative to scFv in CAR T-cell therapy. Previous preclinical study reported in vivo validations of CAR with VH only domain against CD33 that can inhibit tumor growth in mice and prolong survival rate. The remaining tumor cell was lower and more CAR T-cell number after treatment. Also, VH only anti-BCMA showing similar anti-tumor functions compared to their scFv CAR. In this study, we focus on folate receptor alpha or FR-alpha. FR-alpha has been used as target for cancer treatment using various platforms such as folate or antibody drug conjugated, monoclonal antibodies, CAR T-cell targeting folate receptor through scFv called tumor cell killing, and folate antibody nanoparticle conjugated. Using bioinformatics online tool, we found that FOLR1 genes has been detected to be overexpressed in many solid tumors such as cholangiocarcinoma, glioblastoma, ovarian, pancreatic, and endometrial cancer. And high operations of FR-alpha is associated with short survival in ovarian cancer. A human variable domains of VH antibody fragment specific FR-alpha has been previously developed using phased biopanding method by our collaborator. In this study, we established the second generation CAR that were constructed by adding FR-alpha VH sequence derived from phase display of human library. scFv CAR T cells targeting FR-alpha was used as positive control. We also add the CD19 marker to transduction efficiency detection. The transduction efficiency of our CAR was about 30 to 40% after transduction for 4 and 11 days. T-cell patients and absolute CAR number did not differ significantly between scFv and VH CAR. For immune characterizations, our CAR products show 2-to-1 ratio of CD8, CD4 T cells. VH CAR T cell have higher number of central memory T cell, but scFv CAR show higher number of naive stem-like memory T cells. In this study, we use ovarian cancer cell line SKOV3, CAOVT as folate receptor expressing target cell, and largely were also used as negative target cell line. To study the functions of our CAR products, the anti-FR-alpha CAR were cultured with target cell at different ET ratio for 3 days. The results show that both CAR can kill FR-alpha positive target cell in a dose-dependent manner, but no effect on negative target cells. SCFV can show significant increase CD25 and CD69 activation marker comparing with VH-derived CAR T cells after co-culture. In addition, SCFV CAR T show higher level of interferon gamma, TNF alpha, interleukin 2, interleukin 4, and interleukin 10 secretions compared to VS CAR products. Interestingly, after co-culture with SKOV, CAOV3 cells, VS CAR T cells have less exhaustion level of exhaustion marker, includes PD-1, TIGIT, and RAC-T compared to SCFV CAR T products. Moreover, VS CAR show more proliferating cells. CD3 positive were gated to exit adenosine 5 staining for apoptosis analysis, which results show that VS CAR products have less number of apoptotic cells versus SCFV CAR. In summary, in this result provide the proof of concept for human VH-based CAR T target FR-alpha for treatment the solid tumor. We successfully developed a second generation of CAR target FR-alpha from human VH antibody. Both CAR show specific killing effect on FR-alpha ovarian cancer in a dose-dependent manner. VH CAR have less exhaustion and increased proliferations after co-culture. Also, SCFV CAR show higher cytokine secretions and more activations induce cell death. For the further experiment, we plan to perform repetitive tumor re-challenge in vitro and long-term 3D spheroid culture to predict anti-tumor function of our CAR. And phenotype after co-culture will be investigated to explain higher proportions of activation and cytokine release in CAR T cells. Moreover, the anti-efficacy of this CAR will be confirmed in animal model before doing clinical trial. I really appreciate for the supportive feedback our professor have provided me in this project. Also, thanks to all CUCAT lab members. Thank you.
Any question?
Great job, thank you. So did you check the affinity between the SCFV and VH-only CARs to the target?
Affinity.
The affinity.
Affinity.
You use the same VH. You use the VH from the SCFV, right?
Not.
No.
Yeah. For now.
Yeah.
No.
So you use a different VH from the SCFV. And what is the differences of the affinity between the VH-only CARs and the SCFV to the target?
Oh, I also plan to do later.
Okay.
Thank you for your talk. I'm here. Yeah, I would like to ask a question. So you talk about that your target is expressed in a lot of solid tumors. And what about the normal cells? So because folate is something that is very common in a lot of cells, did you look where it is expressed not in cancer cells? And is it dangerous?
For now, I did not investigate in the normal cell, but that question is very important to before use it in humans. Thank you.
So I'm upset that, can you go back to the expansion graph? So on day 4 and day 11, after expansion, day 4 and day 11, I observed that the positive CD19 percentage is decreasing. After expansion, what is the mechanism why the CAR expression is decreasing? Because you use the CD19 for the marker, right? This means that is the percentage of CAR positive cells. So after expansion, day 4 and day 11 is to decrease the number of CAR in your sample. What is the mechanism? Why is die or the percentage is decreased? It should be like persistence if it can grow along with the normal and transduced T cells. Transduce CAR structure ค่ะแล้วก็หลังจาก day 4ค่ะเปอร์เซ็นต์มันประมาณ 44 กับ 46%หลังจากนั้นนะคะเลี้ยงไป 11 วันเนี่ยมันลดลงเหลือแค่ 30% นั้นคำถามก็คือว่า CAR เนี่ยที่มันมี structure มันตายหรือเปล่าทำไมเปอร์เซ็นต์ของ untransduced มันถึงเพิ่มขึ้นพวกนี้มัน toxic หรือเปล่าหรือว่ามัน kill เป็น fratricide หรืออะไรอย่างอื่นหรือเปล่าคะทำไมมันถึงน้อยลง
for fratricide อะครับผมเช็คแล้วก็คือเปอร์เซ็นต์a positive ไม่ได้แตกต่างกันครับ
แมคานิซึมอื่นนะคะอย่างเช่นพวก signaling ที่มันมากเกินไปบางทีพวกนี้ก็ทำ over activation พวกannexin 5 staining หรืออะไรอย่างอื่นก็ได้ค่ะคือทำไม CAR positive มันถึงน้อยลงคือถ้าเกิดว่าเราจะ application พวกนี้ค่ะคืออันนี้มันแค่11 วันถ้า prolong เรื่องของ persistence ของ CAR T มันก็สำคัญนั้นเนี่ยถ้าเกิดว่าT เซลล์ที่มันมีตัว structure ค่ะมัน dying อย่างเงี้ยค่ะมันก็จะไม่น่าจะโอเคสำหรับเรื่องของ treatment application ต่างๆอะไรเงี้ยค่ะ
แต่ for baseline ที่ day 11นะครับก็ลองเช็คพวก annexin 5 positive แล้วมันก็ไม่ได้แตกต่างกันในทั้ง2 construct ครับ
Hey, hi, nice talk. So in your cell killer assays, right, you use a 72-hour time point for evaluation. 72-hour time point for evaluation of cell kill. Why do you use that long to co-incubate? Do you have data for 24 hours as well?
Oh, I did for short term 24 hour.
So it didn't show up in 24?
It can kill target cell too.
All right, for the interest of time, I think we need to cut this session now because Professor Takashi is waiting for us inside the Zoom meeting. So, thank you for all the presenters of this first oral session.